So today we are going to start by just finishing up some general chemistry terms, acids and bases, a little bit about water and its unique properties as a compound. Then we'll start to get into the biological macromolecules. So first, to start with acids and bases. So the acidity or the pH of a solution is a characteristic that we often measure. And so you learned a little bit about water yesterday when we were doing that packet. And one of the things that you saw is that water is a polar molecule. The parts of the water molecule tend to have a positive charge, other parts have a negative charge. So when water is added to ionic compounds that have ionic bonds, often those bonds separate and the two ions dissociate from each other. They separate from each other. Okay? Water, when it dissociates, so H2O, forms two different ions. Okay? One is a hydrogen ion, H+, and then the other is a hydroxide ion, OH-. And the relative proportion of those two ions in a solution is what determines its acidity, its pH. Pure water has equal amounts of hydrogen ions and hydroxide ions. Okay? So these brackets mean concentration in chemistry. So pure water that's neutral in its acidity has equal amounts of hydrogen ions and hydroxide ions. In acidic solutions, there's a greater proportion of hydrogen ions. Okay, so more hydrogen ions leads to an acidic solution. The opposite of an acid is a base. The opposite of acidic is basic or alkaline. In those solutions, it's the opposite. There are more hydroxide ions than there are hydrogen ions. And so if we combine acids and bases, okay, that can neutralize. Okay, when we add an acid and a base, okay, those hydrogen ions, those hydroxide ions can recombine and form water, okay, balance each other out, and return the solution to neutral. So as an example, here's a reaction. Hydrochloric acid, it's a pretty strong acid. Sodium hydroxide can be a strong base. When you mix hydrochloric acid and sodium hydroxide together, the hydrogen ions and the hydroxide ions combine to form water. And then the two other elements, sodium and chlorine, they combine and form a salt. In this case, sodium chloride. So that neutralization tends to bring the solution back to a neutral pH.
Wait, so did the oceans like that, or did they?
The oceans, there are salts in the oceans in different solutions of different types of minerals, and it varies depending on the location and what part of the ocean it is. But seawater is slightly basic.
Oh, okay.
Let's see. Oh no, that's not what I wanted. All right, so the scale that we use to measure acidity is called the pH scale. And you guys heard about it a little bit. It's a scale that was developed that sort of specifies the pH, or specifies the acidity or the alkalinity of a solution. It was developed in the early 1900s. It stands for potential hydrogen, pH. The pH scale goes from 0 to 14, with 7 in the middle. 7 indicates neutral. And the lower the pH number, the more acidic the substance is. The higher the pH number, the more basic or alkaline it is. So if we look at some common substances you might be familiar with. So 7 is neutral. Milk, just slightly acidic at 6.5. Coffee is acidic at 5. So, sometimes people have the mistaken idea that, oh, if something is acidic, it's like going to burn your skin if you spill it on you. And there are some strong acids that are so strong they could burn your skin, but obviously milk or coffee, they are acidic, but it doesn't mean that they are damaging. Beer, lemon juice is pretty acidic as well. Gastric acid from our stomachs, battery acid that's in car batteries, very acidic. So once we start getting into this range down here, those could be dangerous acids that could actually burn your skin.
Well, gastric acid does not feel very good
Yeah.
when you throw up.
Yeah, and another thing is, so people that have various health issues that they vomit very frequently, actually the constant presence of acids from your stomach can start to erode tooth enamel, okay? Can start to damage the inside of the esophagus. People that have chronic acid reflux, where gastric juices and acid is creeping into their esophagus all the time, it can wear away at the lining of their esophagus and cause health problems. So it's why people that have heartburn every day, heartburn's caused by that stomach acid going up your esophagus, should control it using a medication so it doesn't do that damage. The opposite end of the scale, the basic substances, alkaline substances, our blood is slightly alkaline, okay? Seawater is slightly alkaline, hand soap, ammonia, bleach, lye is very basic. So these things, bleach or ammonia or lye, these things can be damaging as well. If you got them on your skin or in your eyes, strong bases can be just as damaging as strong acids. Typically, if you got a strong base or a base on your hands, it feels slippery. If you ever are cleaning something with bleach or ammonia or something, it feels slippery on your skin. Rick, did you have someone have a question?
You answered it.
Okay. So the pH scale actually, each one increase or decrease in the pH scale actually represents a 10 times increase in acidity or in alkalinity. So something that is a pH of 3 versus something with 2, the 2 is 10 times more acidic. And 1 is 100 times more acidic than 3. And 0 would be 1,000 times more acidic. So it is an exponential scale.
Is there anything that's a 0 for acidity?
0?
Yeah, anything that's a 0?
Not that I know of. All right, so we have sometimes a variety, we sometimes need to measure the pH of a substance, and there's different ways that you can do that. One way is we have pH probes that we'll use in a lab this unit, in which they measure the pH digitally and give you a display. But there are also indicators that we can use. I'll just show you some of these indicators here. All right, so what I have here just to show you some of these things, this is sodium hydroxide.
Yep.
What is that? No,
The base.
it's a base. Sodium hydroxide is a base. This is HCl.
Hydrochloric acid.
That's hydrochloric acid. So these are acids and bases. So obviously they have various pHs. And let's see, phenolphthalein. Phenolphthalein is this indicator. Okay, it is, it's clear. It starts off clear. And it stays clear when it is at a pH of less than 9. So from slightly basic to acidic. Okay, but if you add to that phenolphthalein okay, you'll see at first, okay, until the pH changes significantly. Let's see. Could probably this. Sometimes you do have to test the water in your pool. So if I just add a little bit more, I'll probably bring the pH up above 9, and you can see that change.
It's kind of like an orangey scarlet red.
Yeah. Let's see. Let's go a couple more. Now, could I change this back to clear?
I believe so.
Yes.
Yeah, so now this solution that's turned purple, okay, is basic. But if I add some hydrochloric acid to it, that hydrochloric acid will do what to the base?
It'll bring it down.
Neutralize it, and should be able to get back to clear. So that's phenolphthalein. That's an indicator that indicates basic conditions. Bromothymol blue is another one. This is bromothymol blue.
Bromothymol. Whoa, that's interesting.
So I'm going to add I'm going to dilute it a little bit because it's really concentrated. This is just some distilled water. All right, so this is bromothymol blue.
And it's blue.
Yep, it is blue. And it starts off blue, and under basic conditions stays blue. But when you add acid, it indicates acidic conditions. Let's see if we can.
Whoa.
So under acidic conditions, turns yellowish. Okay. Could I turn it back?
Yes.
How?
Adding the base.
If I add some base to it. Now, if you get it right, you can get it to be green. If it's at. And why you see it sometimes start to change and go back, that's because conditions have been neutralized. Is that greenish? That's pretty much blue. All right, so yeah, this is an indicator. It goes back and forth, and you could change it back and forth multiple times. That's bromothymol blue. There's also papers that you can use. This is called litmus paper. Okay, and litmus paper, there's different types. There's red, there's blue, and then there's sort of general pH paper. So red litmus paper starts off red in acidic conditions, turns blue in basic conditions, or you can have blue litmus paper, any guess?
It's the other way around.
In
Acidic condition.
acidic conditions. So, turns red in acidic conditions. We also have this other paper. This is a type of paper that turns a variety of colors. And it has little scales. So this paper, it's called P-hydrin paper. And there's different types you can get. But you put this into a solution, like, let's see, I don't know what the pH of this is, let's see. Put it in there. Okay. Let it dry. And then you compare the color to the colors on the container, and you can tell sort of what pH. This is about 5, so slightly acidic. If I use a different piece, let's put some hydrochloric acid on it. You can see it has a much greater change. Then again, you compare it to this, that's around 1. So this can also give you an indication of the acidity. So these are all indicators that we use to try to assess the pH of a substance. What do you think the pH of my coffee is? Let's try that. So what did our chart said? Coffee's about
5.
I don't want to spoil my coffee. Yeah, pretty close. That's 5. Maybe a little more than 5. I mean, a little less pH-wise. Maybe it's a little below that, but yeah. So my coffee's slightly acidic. All right. Any questions about those indicators? All right. So phenolphthalein, okay, turns, just to review, pinkish violet at higher pHs when things are basic. Bromothymol blue, which goes from yellow when it's acidic to blue when it's basic, and then in between it's at 7, it's greenish. Paper, so red litmus paper or blue litmus paper,
Wow.
and our P hydrogen paper that turns red. All right. So yesterday you read about water a little bit. So water is extremely important to life on Earth, obviously, and it has some unique properties that are really important. So you learned yesterday that water is a polar molecule. If you look at how the electrons are shared between the hydrogen and the oxygen, okay, that the electrons are more sort of concentrated, have a higher density on the oxygen molecule, giving this a negative charge, and then a positive charge at the 2 hydrogens. So that property, where you have part of the molecule is positive and part is a negative charge, gives water some of its unique properties. So water is obviously 2 hydrogen atoms with what we call polar covalent bonds. So they're covalent bonds because the electrons are being shared between the hydrogen and oxygen. They're polar bonds because they're not shared equally. The electrons are being pulled closer to the oxygen, making the molecule have a charge. And because of that, water molecules can form these weak hydrogen bonds between each other. You were drawing them yesterday with dotted lines, showing the oxygen and hydrogen forming these weak hydrogen bonds. And because of those weak hydrogen bonds, water has some properties like cohesion. Water molecules like to stay together. They have those hydrogen bonds keeping them together. Back in 7th grade, you guys did that experiment. How many drops can you put on a penny? The reason, though, you could add all those drops and they form a little dome of water is because all the water molecules have these weak hydrogen bonds keeping them together. So in 7th grade, we said the water molecules are sort of sticky with each other and compared them to like a pile of tape balls. That you can pile up balls of tape and they stay together because they're kind of sticky, but at some point, force of gravity overwhelms the stickiness. At some point, the force of gravity pulling on those water molecules overwhelms the weak hydrogen bonds and they no longer hold together. Water also has surface tension, where the surface of water is the molecules have weak bonds, which makes it more difficult to break through them. This is how insects can walk on top of the water, because the force of them walking on the water is not great enough to break those hydrogen bonds, and so they walk right on top of the water.
And is that also why it hurts whenever you jump into the puddle?
Yeah, that has something to do with it, I guess, yeah. Never thought of it that way. Water also has a high specific heat. It takes a lot of energy to change the temperature of water. It resists changes in temperature. That's one of the reasons why coastal areas have more moderate temperatures, because the water around them resists quick, large changes in temperature. It's also why when sweat evaporates from your skin, it helps cool you down because it's absorbing a lot of energy, okay, to change into a gas. And water is an excellent solvent. It can dissolve many substances, not everything, but many solvents. It can dissolve many different substances. And one of the reasons it can dissolve many substances is because it's a polar molecule. It has that charge. So we're going to start to get into now some specific organic molecules, classes of organic molecules, and talk about them. Now all of these are organic molecules, therefore what do they contain? Carbon and hydrogen. So, carbon is a very versatile molecule. It has 4 bonds for every atom, as we looked at in that model I showed you the other day. So there can be a huge variety of shape, huge variety of connections between molecules. And so here's some examples. You could have different lengths of different number of carbons bonded together to form different lengths of molecules. Carbon can form double bonds between each other. So sometimes the position of that double bond can be in different places, and that changes the molecule. Carbon can form branches. Instead of being the carbons on a straight line, they can branch off. And that gives a wide variety of molecules. Carbon can form rings connecting with each other, which gives variety. So there's all sorts of shapes and configurations of these organic molecules that are possible. And we are going to talk about 5 main classes of organic compounds: carbohydrates, lipids, proteins, nucleic acids, and other high-energy organic molecules. We'll take them one at a time. And so we're going to start with carbohydrates. So carbohydrates include things like monosaccharides, disaccharides, polysaccharides. And monosaccharides combine to form disaccharides, and disaccharides combine to form polysaccharides. We'll talk about what all those mean. Lipids include things like fats and oils. Proteins include a huge variety of polypeptides. Nucleic acids include DNA and RNA. And then these other high energy molecules are things like ATP. So we'll start with carbohydrates. What is, you learned this in health, I'm sure. What are carbohydrates used for in our bodies?
Energy.
Yeah, energy is a primary function. A gram of carbohydrate provides 4 calories of energy that our body can use. They compose some of our cell structures as well. Some examples of carbohydrates: sugars and starches. The name itself, carbohydrate, carbo refers to carbon because they have a backbone of carbon. Hydrates, water, because they have the other 2 missing elements are hydrogen and oxygen. Many times carbohydrates are named with the suffix OSE: glucose, fructose, maltose, lactose, cellulose. These are all carbohydrates.
Wait, so
Saccharides
are kind of sugars?
Yeah, they're simple sugars, we sometimes call them. One way to identify a carbohydrate is it has a consistent ratio of 2 hydrogen to 1 oxygen. Imagine glucose, C6H12O6. So 12 hydrogen to 6 oxygen. There's that 2 to 1 ratio. So the simplest carbohydrates are the monosaccharides. They are just the building blocks, the subunits, we call them, of larger carbohydrates. Next are disaccharides, which are made of 2 monosaccharides. And polysaccharides have many of these monosaccharides bonded together. So let's talk about first the monosaccharides, mono meaning 1. So the formula for monosaccharide is always C6H12O6. Now we always call that what? glucose, but it can also be other things. It can be fructose, it can be galactose. So how could the same formula be those three different things? Anyone have any idea? It's all C6H12O6. Will it?
Different structures?
Different structures, different rings, different ways that those C6H12O6 atoms are connected to each other.
So, monosaccharides combined to make disaccharides. Is it two of the same monosaccharides?
It can be, or it can be two different. So often these monosaccharides form a ring shape, and we might draw them as a hexagon or a pentagon because they form that ring. So when you see that shape, you're thinking it's a monosaccharide. So examples: glucose, fructose, and galactose. They are all C6H12O6. They're isomers of each other. Glucose is obviously really important. You learned about it in cellular respiration, photosynthesis. It is one of the prime raw materials for cellular respiration. Fructose is produced in fruits, okay, in plant nectar, in honey. This is one of the sweetest, in terms of taste, monosaccharides. It's why high fructose corn syrup is used often as a sweetener in food products. Galactose is another monosaccharide, and it moves through the cell membrane pretty quickly. It's a component of larger polysaccharides, and so they're all C6H12O6. Here are what their structural formulas look like. So you have fructose, which has this pentagon ring shape, glucose, and galactose, which have that hexagon. And you can see there's slight differences in where the hydroxide group is between glucose and galactose, and that's what makes them different from each other. Here's what they look like if we're to draw this structural formula. Okay? So again, they're all isomers. All right, so disaccharides are two of these monosaccharides combined. They're double sugars, di meaning two. So if we have these two hexagons connected by an oxygen, that would indicate a disaccharide. Some examples of disaccharides you might be familiar with are maltose, lactose, sucrose. Maltose is found often in germinating seeds. It's created by the digestion of starch. And maltose is two glucose molecules bonded together. So two monosaccharides of glucose bonded together form a maltose. Lactose is found in dairy products often. We started talking about that. We'll talk more about it. Lactose is a galactose monosaccharide combined with a glucose. Sucrose is table sugar, stuff you have in your kitchen. Okay? And sucrose is a combination of a fructose and a glucose combined. So these are all disaccharides. We can tell they're two ring shapes bonded together by an oxygen. And then if we continue to add more monosaccharides together, we form polysaccharides. These can be many hundreds or even thousands of monosaccharides bonded together. These are large molecules typically. So we would kind of draw them with a series of hexagons connected with oxygens in between. Some examples: starch. Starch is a bunch of glucose monosaccharides bonded together. Starch is used for plants to store energy. Cellulose, which makes up plant cell walls, is another polysaccharide. This is cellulose. It forms structural components. It's strong and durable. Chitin forms the exoskeleton of insects and the cell wall of fungi. It's another polysaccharide. Glycogen is how our body stores energy in our liver. It's a polymer of glucose as well, but a very complex molecule. So how do these monosaccharides combine? Through a process called dehydration synthesis, which if you think about the words, dehydration means to lose water, synthesis means to build up a larger molecule. So two monosaccharides, for example, I have two monosaccharides here, can be joined together through dehydration synthesis. This is showing maltose being formed from two glucose molecules. Enzymes allow this to happen. And so what happens here with these two glucose molecules is the hydroxide of one gets combined with a hydrogen from the opposite hydroxide group and gets released as water. And what's left here is oxygen, and that gets bonded to the carbon. So you see in here, we formed a disaccharide from two monosaccharides, and the product that's released is water. So that's called dehydration synthesis. The opposite process is called hydrolysis. Hydro meaning water, lysis meaning to break down. And it's the opposite basically. You add water to a disaccharide, this is sucrose. If we add water, okay, an OH gets bonded to one of the monosaccharides, a hydrogen gets bonded to the oxygen, and now we've broken down a disaccharide to form two monosaccharides. It's the opposite.
So this is how, basically how, with dehydration synthesis is how two monosaccharides become disaccharides?
And hydrolysis is how a disaccharide gets broken up into two
okay.
monosaccharides.
Ah, all right.
And repeat that process, we can build polysaccharides by repeated dehydration synthesis, or we could break down a large polysaccharide into monosaccharides using what process? Hydrolysis. Okay. All right. Any questions about carbohydrates or dehydration synthesis and hydrolysis?
Wait,
Need the notes there, or you want to look at the diagram?